Okay, then let's start with class 12 biotechnology principles and processes. But before that, there has been an introduction given here. Let's see that. Unit 9 Biotechnology. This includes two chapters: Chapter 9 Biotechnology Principles and Process, and Chapter 10 Biotechnology and its Applications. Ever since the days of René Descartes, the French philosopher, mathematician, and biologist of 17th century, all human knowledge, especially about natural sciences, were directed to develop technologies which add to the creature comforts of human lives, as also value to human life. The whole approach to understanding the natural phenomena became an anthropocentric. Physics and chemistry gave rise to engineering, technologies, and industries which all work for human comfort and welfare. The major utility of the biological world is a source of food. Biotechnology, the 12th century offshoot of modern biology, changed our daily life as its product brought qualitative improvement in health and food production. The basic principles underlying biotechnology possesses and some application and highlighted and discussed in this chapter. Okay, ever since René Descartes, he is a French philosopher and mathematician, biologist of the 17th century, meaning he lived in the 1600s. All human knowledge, especially natural sciences, were directed to develop technologies to add to feature comforts of the human life. Everything, every technology, every natural sciences we pursue is everything is for comforts of the human life only. The whole approach to understanding natural phenomena became anthropocentric, meaning is humans as the center. Why we understand and go through all this is because of more comfort and more ways of us surviving, etc. Physics and chemistry gave rise to engineering, technologies, and industries which all worked for human comfort and welfare. So many companies which were given rise to by physics and chemistry. So many branches, but most of them, all of them are for human comfort and welfare. As for we see many of those companies. And the major, like what is the utility of the biological world is that biological world is a source of food. Biotechnology, the 20th century offshoot of modern biology, changed our daily life as its product brought qualitative improvement. Biotechnology changed our daily life as its products brought qualitative improvement in health and food production. It brought qualitative improvement in health and food production. The basic principles underlying biotechnological processes and some applications are highlighted and discussed in this unit. What we discuss in this unit is basic principles underlying biotechnological process and some applications are discussed in this unit. Now let's start with chapter 9, that is biotechnology principles and processes. There is something about someone given here. I'll just read it out. Herbert Boyer was born in 1936 and brought up in a corner of western Pennsylvania where railroads and mines were the destiny of most young men. He completed graduate work at the University of Pittsburgh in 1963, followed by 3 years of postgraduate studies at Yale. In 1966, Boyer took over assistant professor at the University of California at San Francisco. By 1969, he performed studies on a couple of restriction enzymes of the E. coli bacterium with especially useful properties. Boyer observed that these enzymes have the capability of cutting DNA strands in a particular fashion. Restriction enzymes. These enzymes have the ability of cutting DNA strands in a particular fashion, which left what has become known as sticky ends. And the strands, these clipped ends, made pasting together pieces of DNA a precise exercise. We always think how we know that DNA is the blueprint of life. So, what if we can take DNA from our desired animals and integrate into our own genes? Can we run like a cheetah if we take its gene? That is not exactly possible right now, but that is exactly what he is trying to do. Cutting the genes and taking that exact DNA and keeping it in our needed DNA so that new modifications are seen. This cutting is not done by physical scissors, but uh, enzyme scissors here, that is restriction enzymes, so that we get a precision in the cutting of the DNA and pasting it. This discovery in turn led to rich and rewarding conversation in Hawaii with Stanford scientist Stanley Cohen. Cohen and had been studying small ringlets of DNA called plasmids and which float about freely in the cytoplasm of certain bacterial cells and replicate independently from the coding strand of the DNA. Okay, what this Cohen scientist, Stanley Cohen, a Stanford scientist, what he was observing was there were small ringlets meaning ring-shaped things like this, ringlets of DNA. These were called as plasmids, which float about freely in the cytoplasm of certain bacterial cells. They float around in the cytoplasm. They are not present in every cell. They are present in certain bacterial cells. And what is the property that is worth studying is that they replicate independently, replicate independently. That is different from the coding strand of DNA. Normally, the DNA replicates using the semi-conservative nature of DNA, but that is different from what the plasmids do. They replicate independently of the DNA coding strand. Cohen had developed a method of removing these plasmids from the cell and reinserting them in other cells, removing from the cells which contain it and add them to other cells. Combining like if these plasmids we know they are self-replicating, and if we add the DNA to these plasmids and we take out these new mutated plasmids or altered plasmids, they have the DNA, and if we inject them into our desired cells, they will continue to replicate. The cell DNA will also continue to replicate, and because there is an existence of our altered DNA within the plasmids, there is altered DNA within the plasmids. The new phenotypic R desired trait will even show in the new cells. Combining this process with that of DNA splicing enabled Boyer and Cohen to recombine segments of DNA in desired configurations and insert DNA in bacterial cells. which could then act as manufacturing plants for specific proteins. This breakthrough was the basis upon which the discipline of biotechnology was founded. This is what they came basically. Uh, Boyer first found out about restriction enzymes, which had the capability of cutting DNA strands at a particular and precise fashion, and something known as sticky ends, the clipped ends, which made pasting together pieces of DNA precise exercise. And Cohen, who was studying about plasmids, small ringlets of DNA that float freely in the cytoplasm of certain bacterial cells and replicate independently from coding strand DNA, and they both they both combined this plasmid and DNA and thought that injecting into bacterial cells will lead to will lead to production like manufacturing plants for specific proteins. This is the breakthrough at the basis which discipline of biotechnology was founded. Let's go to the chapter 9, biotechnology and principles and processes. In this chapter the topics are principles of biotechnology, tools of recombinant DNA technology, processes of recombinant DNA technology. That are the three topics: principles of biotechnology, tools of recombinant DNA technology, and and process of recombinant DNA technology. Recombinant is basically our altered DNA. Now let's start with the lesson. Biotechnology deals with techniques of using live organisms or enzymes from organisms to produce products and processes useful to humans. Okay, what is biotechnology? Biotechnology deals with the techniques of using live organisms or enzymes from organisms to produce products and processes useful to humans. What we use is live organisms, enzymes, or that live organisms itself to get products which are useful to humans. That is what we call anthropocentric because it is surrounded around humans. In this sense, making curd, bread, or wine, which are all micro-mediated processes, could also be thought to be as a form of biotechnology. What biotechnology deals with is a technique that is used to use living organisms or their enzymes to create products or processes useful for human beings. If that was the case, production of curd, which is bacteria mediated, along with bread fermentation of wine, bread, etc., is also they use yeast here, and for curd they use lactobacillus. This is also a kind of biotechnology process because we are using bacteria for this bacteria as a organisms to produce products useful to humans, that is bread, wine, and curd. However, it can be perceived like that. However, it is used in a restricted sense today. Just because of that, we do not call that as biotechnology. It is more of a restricted sense today. Like we only use it for some things, what we will see, to refer to such of those processes which use genetically modified organisms to achieve the same on a larger scale. Biotechnology is not only what this above definition is, we even though it is correct to call formation of or production of curd, bread, and or wine as biotechnology, we do not use it for that. Biotechnology is specifically used for when Genetically modified organisms achieve the or do the process in much larger scales. Refer to process which genetically modified organisms to achieve the same on a larger scale. Further, many other process techniques are also included under biotechnology. There are many processes techniques included under biotechnology, and one of them is if genetically modified organisms can bring about good results, then we call it as biotechnology. For example, in vitro fertilization leading to a test tube baby synthesizing a gene and using it, developing a DNA vaccine, or correcting a defective gene are all a part of biotechnology. I'm not sure how in vitro fertilization leading to a test tube baby is a part of biotechnology. Let's ask. It's not using any other organism here, so is it called? It's not genetically modified either. It seems it is because of human benefit, tissue and culture methods, use of laboratory techniques, artificial manipulation of biological activities. Okay. The European Federation of Biotechnology (EFB) has given a definition of biotechnology that encompasses both traditional view and modern molecular biotechnology. The definition given by EFB is as follows. Basically, the European Federation of Biotechnology, represented by EFB, had given a definition of biotechnology. This covers both traditional view of biotechnology and modern molecular biotechnology. The definition given by EFB is as follows: the integration of natural science and organisms, cells, parts thereof, and molecular analogs for products and services. The integration of natural science and organisms, cells, parts thereof, and molecular analogs for products and services. Okay, that is the definition given by EFP. This is for products and services. Here we are integrating natural science, organisms, their cells, their parts, molecular analogs, that is for products and services. Now let's go to the first main topic, that is principles of biotechnology. Principles of biotechnology, among many, the two core techniques that enabled birth of modern biotechnology are there are many techniques that birth the modern biotechnology, but the main two are given here, I believe. Yes, they gave only two. That is, the two are genetic engineering and bioprocess engineering. Let's see what they are. The two core techniques that enabled the birth of modern biotechnology are genetic engineering, techniques to alter the chemistry of genetic material, DNA and RNA, to introduce these into host organism and thus change the phenotype of the host organism. genetic engineering techniques to alter the chemistry of genetic material, DNA and RNA, to introduce this into host organism and thus change the phenotype of the host organism. Okay, what this genetic engineering is, as the name suggests, genetic engineering, we basically alter the chemistry of genetic material, we change the structures, we change the chemistry of genetic material that can be either RNA or DNA, and this altered DNA we introduce into the host organism, and we expect to see changes in the phenotype of the host organism because we inserted new DNA. Now let's go to the second technique. First is genetic engineering, which is very obvious. Bioprocess engineering, maintenance of sterile, microbial contamination free ambience in chemical engineering processes to enable growth of only the desired microbe eukaryotic cells in large quantities for the manufacture of biotechnological products like antibiotics, vaccines, enzymes, etc. Okay, the second technique is bioprocess engineering, maintenance of sterile ambience in chemical engineering process to enable growth. These are the four steps. That is upstream processing, preparing culture media, optimizing nutrients. Choosing a culture media is for bacteria basically. Bacteria need the food and nutrition to grow, so it needs specific culture media, that is and optimizing the nutrients for the specific bacteria. Choosing and maintaining microbial cell cultures basically just colonies of bacteria. Maintaining a sterile environment, not about these bacteria but other species that might invade and contaminate, keeping only the wanted microbe alive. And next is bioreactor design and operation, scaling up from lab flask to large fermenters, controlling temperatures, pH, oxygen, agitation, etc. We should control bioreactor design and operation. Production of useful products. Microbes of cells are used to produce antibiotics, vaccines, hormones like insulin, enzymes, alcohol, etc. This includes drug production through microbes, but also extends to biofuels, food production, and even bioplastics. Downstream processing is separation, purification, and formation of product. For example, purifying insulin from E. coli cultures. Okay. Is a maintenance of sterile microbial contamination-free ambience in chemical engineering process to enable growth of only the desired microbe eukaryotic cell in large quantities for the manufacture of biotechnological products like antibiotics, enzymes, etc. Let us now understand the development. Let us now understand the conceptual development of the principles of genetic engineering. You probably appreciate the advantages of sexual reproduction over asexual reproduction. The former, that is sexual reproduction, provides opportunities for variations and formulation of unique combination of genetic setup, some of which may be beneficial to the organism as well as the population. Asexual reproduction preserves the genetic formation. Okay, reproduction preserves the genetic information, while sexual reproduction permits variation. Traditional hybridization process used in plant and animal breeding very often lead to inclusion and multiplication of undesirable genes along with the desired genes. Okay, let's see what they're saying here. The traditional hybridization process or procedure, the same hybridization or genetic alteration if we do in plants and animal breeding, very often this lead to inclusion and multiplication of undesirable genes along with the desirable genes. The techniques of genetic engineering, which include creation of recombinant DNA, use of gene cloning and gene transfer. The techniques of genetic engineering, which include creation of recombinant DNA. Use of gene cloning and gene transfer. Normally, the limitations we see with the hybridization we see in animal and plant breeding, this limitation is that the inclusion and multiplication of undesirable genes. Even though the desired genes are multiplicating, even the undesired genes are multiplicating. But to avoid this, the technique of engineering, which includes creation of recombinant DNA, altered DNA, we alter the DNA, use of gene cloning and gene transfer, that is biotechnology. Using these techniques, using creation of recombinant RNA, technique of gene cloning and gene transfer, we overcome this limitation and allow us to isolate and introduce only one or a set of desirable genes without introducing undesirable genes into the target organism. Basically, if normally in the olden days, when we wanted a new breed, we, for cows example, we used to breed between two different species of cows or two different types of cows. But there might be many desirable characteristics in the both cows. There are also undesirable traits as well. But we only want desirable traits to multiply, not the undesirable ones. So breeding is not through breeding, like through selected breeding. Even though might get most of the desired characters, we will also get most of the unwanted characters as well. But with techniques of genetic engineering, that is to recombinant DNA, we don't need to worry about it. We can individually select which character we want. That is by taking the DNA using restriction enzymes and cutting our specific DNA and using plasmids to attach this cut DNA and make it self-replicate and insert into the host organism. That is recombinant DNA. recombinant DNA with use of gene cloning and gene transfer. We overcome that limitation of breeding and we can get our desired trait by ourselves. Only one set of desirable genes without introducing undesirable genes into the target organism. Do you know the likely fate of a piece of DNA? Do you know the likely fate of a piece of DNA which is somehow transferred into an alien organism? Most likely, this piece of DNA would not be able to multiply itself, the progeny of cells of the organism. Okay, we already know that our body treats foreign particles very carefully, and that is that is the reason we are able to survive. It's our immunity power. And similarly, just because we add a piece of DNA, I'm only talking about without plasmids. Just let's assume we take a piece of DNA using restriction enzymes and just place it in a new organism, just like that, in an alien organism. Most likely, this piece of DNA would not be able to multiply. How will it multiply? It doesn't have any mechanisms. It's just a segment of DNA. It will not multiply in the itself in the progeny of the cells of the organism. But we can make it replicate if but when it gets integrated into the genome of the recipient, it may multiply and be inherited along with the host DNA. This is because the alien piece of DNA has become a part of the chromosome, which has the ability to replicate. Okay, let's not talk about plasmids and all. Most obvious idea, if we can't just directly take a DNA and insert into a cell, then most obviously we should think that we should add this into the cell genome itself where chromosomes are there. If we add it to the already existing DNA, attach it there, then because it will become a part of a chromosome which have the ability to replicate, then it will continue to replicate along with the chromosomes. Then the DNA that is completely transferred and completely altered, our specific gene has been introduced. Now let's see. In a chromosome, there is a specific DNA sequence called the origin of replication, which is responsible for initiating the replication. Therefore, for multiplication of any alien piece of DNA in an organism, it needs to be a part of a chromosome, which has a specific sequence known as origin of replication. Okay, information given here is okay, in a chromosome, there is a specific DNA sequence, and this sequence is called as origin of replication. This is responsible for initiating replication. Therefore, for multiplication of any kind of alien piece of DNA in an organism, it needs to be a part of the chromosome, which has a specific sequence known as the origin of replication. Thus, an alien DNA is linked with the origin replication so that this alien piece of DNA can replicate and multiply itself in the host organism. This can also be called as cloning or making multiple identical copies of any template DNA. Known as like chromosomes have a sequence called as origin of replication. So if we attach our alien DNA to that part, it will continue to replicate and we will get a desired trait or gene. We will get a desired phenotype. And DNA can replicate and multiply itself in the host organism if it is attached to the chromosome's origin of replication site. That this can also be called as cloning or making multiple identical copies of template DNA. This process of multiplying itself in the host organism is called as cloning. Let us now focus on the first instance of the construction of an artificial recombinant DNA molecule. The construction of the first recombinant DNA emerged from the possibility that of linking a gene encoding antibiotic resistance with a native plasmid. Okay, now let us see the first instant, the first time, the construction of an artificial DNA recombinant molecule. The construction of the first recombinant DNA emerged from the possibility of linking a gene encoding antibiotic resistance with native plasmid. Linking a gene encoding antibiotic resistance with a native plasmid. This plasmid is a autonomously replicating circular extrachromosomal DNA. Okay, of Salmonella typhimurium. What we are doing for the first recombinant recombinant DNA. Recombinant DNA means DNA made by joining from two different sources. The first recombinant DNA refers to very first artificial gene construct made in lab, gene encoding antibiotic resistance. Scientists took a gene that makes bacteria resistant to specific antibiotic, say tetracycline or ampicillin. This gene came from one organism, not necessarily Salmonella. Okay, native plasmid of Salmonella typhimurium plasmid, small circular extrachromosomal DNA found in bacteria. They replicate independently of main bacteria chromosome. Scientists use such a plasmid from Salmonella typhimurium as vector, linking the two using restriction enzyme to cut and ligases to join DNA. The antibiotic resistance gene was inserted into the plasmid DNA. Now the plasmid carried foreign DNA along with its own DNA. The plasmid carrying the antibiotic resistance gene could be introduced into bacteria. The bacteria would gain resistance, providing the foreign gene was functional. This experiment laid the foundation for modern genetic engineering and biotechnology. Example, making insulin, growth hormone, vaccines through recombinant DNA. Okay, basically there is a gene that gives antibiotic resistance, a specific antibiotic resistance. For a specific antibiotic resistance to the bacteria Salmonella typhimurium. Only a specific antibiotic resistance. So we take that gene and insert it into a native plasmid of this Salmonella typhimurium, so that if this bacteria, after adding it, it will become recombinant DNA, meaning The plasmid will contain not only its own DNA but also the DNA, the DNA having the gene coding for a phenotype of antibiotic resistance, that is against one specific antibiotic. It will also have that gene. If after this, the cells of this Salmonella typhimurium now had antibiotic resistance after the experiment. After this recombinant DNA was added, if this bacteria had that specific antibiotic resistance, then it would prove that the plasmids or the recombinant DNA was functional, meaning it was successful, it was working. If it did not, if it did not work, the bacteria still died under that antibiotic, then that means it was not functional. That is the experiment here. Let us now focus the first instance of the construction of an artificial recombinant molecule. The construction of the first recombinant DNA emerged from possibility of linking a gene encoding antibiotic resistance with a native plasmid, autonomously replicating circular extrachromosomal DNA of Salmonella typhimurium. Stanley Cohen, we saw about him before. Stanley Cohen and Herbert Boyer accomplished this in 1972 by isolating the antibiotic resistance gene by cutting out a piece of DNA from a plasmid which was responsible for conferring antibiotic resistance. The cutting of the DNA at specific locations became possible with the discovery of the so-called DNA scissors or molecular scissors, that is restriction enzymes. The cut piece of DNA was then linked with the plasmid DNA. These plasmid DNA act as vectors to transfer the piece of DNA attached to it. Okay, they are acting as vectors just like mosquitoes. You probably know that mosquito acts as an insect vector to transfer the malarial parasite into the human body. In the same way, a plasmid can be used as a vector to deliver an alien piece of DNA in the host organism. Okay, then let's continue. Molecular scissors, restriction enzyme. The cut piece of DNA was then linked with plasmid DNA. The plasmid DNA act as vectors. They act as vectors to transfer the piece of DNA attached to it. You probably know that mosquito acts as an insect vector to transfer the malarial parasite into the human body. In the same way, a plasmid can be used as a vector to deliver an alien piece of DNA into the host. The linking of antibiotic resistance gene with the plasmid vector became possible with the enzyme DNA ligase, which acts on the cut DNA molecules and joins their ends. Restriction enzymes act as molecular scissors, whereas DNA ligase will join up the DNA into the vector. This makes a new combination of circular autonomously replicating DNA created in vitro and is known as recombinant DNA. DNA in vitro and okay, transfer the piece of DNA attached to it. The mosquito. Okay. Escherichia coli, a bacterium correlated to Salmonella, it could a new host's DNA polymer and make multiple copies. Ability of multiplying copies of antibiotic resistant gene in E. coli was cloning of antibiotic resistance gene in E. coli. You can infer that there are three basic steps in genetically modifying an organism: identifying an DNA with desirable genes, introduction of identified DNA in the host, maintenance of introduced DNA in the host, and transfer of DNA to its progeny. Plasmid DNA as a vector. Plasmids are small circular DNA molecules in bacteria separate from the main chromosome. They replicate on their own, autonomously replicating. Scientists used plasmids as carrier vectors to deliver foreign DNA into a host cell. Just as a mosquito carries the malaria parasite into humans, a plasmid carries a foreign gene into the bacteria. DNA ligase, molecular glue. After restriction enzymes cut, the foreign gene is inserted into the plasmid. After restriction enzyme cut, the foreign gene is inserted into the plasmid. DNA ligase seals the end, creating one continuous circular DNA, that is recombinant DNA. Plasmid use the bacterium's normal DNA machinery copy itself. resistance gene, cloning of the gene. Three basic steps of genetic modification generalized. Required DNA is cut and attached to a plasmid and through ligases. This acts as a vector transferring DNA into host as vectors vectors are self-replicating DNA attached to plasmid if the gene were inserted without a plasmid, it would be degraded or lost in the host cell. Okay. Restriction enzyme that cut a piece of DNA was then linked with plasmid DNA. The plasmid DNA act as vectors to transfer the DNA attached to it, like a mosquito act as an insect vector to transfer malaria parasite into a human body. In the same way, plasmid can be used as a vector to deliver an alien piece of DNA into the host organism. The linking of antibiotic resistance gene with a plasmid vector became possible with enzyme DNA ligase, which acts and cut out DNA molecules and join their ends. This makes a new component of circular autonomous replicating DNA created in vitro known as recombinant DNA. When this DNA is transferred into Escherichia coli, a bacteria closely related to Salmonella, it could replicate using the new host DNA polymerase enzyme and take multiple copies. The ability to multiply copies of antibiotic resistant gene in E. coli was called as cloning of antibiotic resistance gene in E. coli. You can hence infer that there are three basic steps in genetically modifying an organism: identification of DNA with desirable genes, introduction of identified DNA into the host, maintenance of introduced DNA of host, and transfer of the DNA to its progeny. This is the result. This was the full experiment, the result of the experiment, that it was successful. Now let's go to tools of recombinant DNA. This is the second main topic, tools of recombinant DNA. Before we saw genetic processing and biotechnology processing, and we saw about that's all. Tools of recombinant DNA technology. Now we know from the foregoing discussion that genetic engineering or recombinant DNA technology can be accomplished only if we have the key tools, that is restriction enzymes, polymerase enzymes, ligases, vectors, and the host organism. Restriction enzymes to cut the DNA, polymerase to clone the DNA, ligases to attach the DNA, vectors to transfer the DNA, and host organism to bear the altered DNA. Let us try to understand some of these in detail. Restriction enzymes. In the year 1963, the two enzymes responsible for restricting the growth of bacteriophage in Escherichia coli were isolated. One of these added methyl groups to the DNA, while the other cut the DNA. The latter, meaning the which cut the DNA, was called as restriction endonuclease. Okay. In the year of 1963, there were two enzymes which were responsible. What they were doing was they were restricting the growth of bacteriophage in Escherichia coli. They were both isolated, these two enzymes. One of these added methyl groups to the DNA, while the other enzyme cut the DNA. The latter enzyme, that is the one which cut the DNA, was called as restriction endonuclease. The first restriction endonuclease, it Hind2, the first restriction endonuclease, Hind2, whose functioning depended on a specific DNA nucleotide sequence, was isolated and characterized 5 years later. The first restriction endonuclease, that is Hind2, whose functioning depended on a specific DNA nucleotide. It was isolated and characterized, that is 5 years later, at maybe 1968. When it was identified first, they did not know the use of it, so it was not named Hind1 and Hind2. And Hind1, the name was given to another restriction endonuclease, which was found after it, but they found the function of it before this one. It was found that Hind2 always cut DNA molecules at particular point by recognizing a specific sequence of 6 base pairs. This specific base sequence is known as recognition sequence for Hind2. Okay, this restriction endonuclease, that is Hind2, its functioning depended on a specific nucleotide sequence, basically base pair sequence. It was found that Hind2 always cut DNA molecules at a particular point. Precision at how it knows at which point is It had a six base pair sequence, specific six base pair sequence, like ATGC, AT, or something like that. This six base pair sequence is specific for Hint2 to take action, is called as the recognition sequence for Hint2. Besides Hint2, today we know more than 900 restriction enzymes that have been isolated from over 230 strains of bacteria, each of which recognize different recognition sequences. Hint2 has a six base pair recognition sequence, and there are 230, I mean 900 other restriction enzymes which are isolated from over 230 strains of bacteria. They also show different types of recognition sequences. They may not be six strains. Even if they are, there are different sequences. The convention of naming these enzymes is the first letter of the name comes from the genus, and the second two letters come from the species of the prokaryotic cell from which they are isolated. Okay, we need to name these restriction enzymes. So how we name them is, the convention is into or anything like that. The first letter of the name comes from genus. First letter in the two it is H, and second letter comes from the species of the prokaryotic cell. Genus and species, just like the binomial nomenclature, but here instead of full names it is just letters. Example is EcoRI. It's also an restriction endonuclease. Comes from Escherichia coli RY13. In EcoRI, the letter R derived from the name of the strain, R strain. Roman numbers following the names indicate the order in which the enzymes were isolated from the strain of the bacteria. Okay, the first letter is, I mean, genus, and the second two letters come from species. If HIN, H is genus name, IN is species name, and R is eco RI or R1. So R1, R is derived from the R strain, and here it might be D is from D strain, and 2. H comes from the genus name Haemophilus. IN, influenzae, denotes the strain of the bacteria, strain D of H influenzae, Haemophilus influenzae. To indicate that it was second in restriction enzyme isolated from the strain E. coli, Escherichia coli, strain R Y13, first enzyme, BAM HI, Bacillus amyloliquefaciens, a large name, strain H, and first enzyme. Restriction enzymes belong to larger classes of enzyme called as nucleases. There are two kinds of nucleases, that is exonucleases and endonucleases. Okay, classification given here. Restriction and enzymes belong to a larger class of enzymes. They belong to a larger class of enzymes. These are called as nucleases. There are two kinds of nucleases, exonucleases and endonucleases. Exonucleases remove nucleotides from the ends of the DNA, whereas endonucleases make cuts at specific positions within the DNA. Okay, exonucleases remove nucleotides from the DNA, ends of the DNA, and endonucleases make cuts at specific positions within the DNA. Each restriction endonuclease functions by inspecting the length of a DNA sequence. Once it finds its specific recognition sequence, it will bind to the DNA and cut each of the two strands of the double helix at specific positions in their sugar phosphate backbones. Each restriction endonuclease recognizes a specific palindromic nucleotide sequence in the DNA. Okay, they didn't give much about exonucleases. All that is given about exonucleases is that it's a part of the nucleases, and these enzymes like cut nucleotides at the ends of the DNA. About endonucleases are enzymes which make cuts in the DNA. They do not cut nucleotides, they cut the DNA directly, they cut the phosphate backbone. They have specific recognition sequences where they will bind to the DNA and cut each of the two strands of the double helix at specific positions in their sugar phosphate backbones. Each restriction enzyme recognizing a specific palindromic nucleotide sequence in the DNA. That is the recognition sequence. Let's see some diagrams given here. Steps in formation of DNA recombinant by action of restriction endonuclease enzyme. Here we are using an enzyme called as EcoRI. The enzyme cut both strands at the same time. EcoRI cuts the DNA between bases G and A when the sequence GAATC is present in the DNA. Okay, this is the plasmid DNA. This is the foreign DNA. Normally, EcoRI has the recognition sequence of GAATGC. I mean GAA GAA T C. This is the recognition sequence, and it makes a cut between G and A only when the sequence GAATC is present in the DNA. If this sequence is present in the DNA, it will make a cut between G and A only. It does not differentiate between foreign DNA or vector DNA. The only thing that matters to it is if it's DNA, if it has a recognition sequence, if it does have the recognition sequence, at that specific site, it will make a cut like this. It will cut like this. These are called as a sticky ends. Sticky ends, they just look like blocks to be attached, perfect for attaching. It will make a cut here like this, and same sequence here for vector DNA. It will make a cut, a slice like this, and thus creation of a sticky ends of the DNA, meaning they can be attached. So they'll get attached just like blocks. It will become a recombinant DNA. Both of its ends will be attached to the vector DNA, and it will become an autonomous replicating DNA. And each restriction enzyme recognizes a specific palindromic nucleotide sequence. It's called as a recognition sequence or a palindromic nucleotide sequence. Do you know what palindromes are? These are groups of letters that form the same words when read both forward and backward. Example, Malayalam. If we go from forward, it is Malayam, and from backward, it is same Malayam. Maybe it is okay. Yes. Palindromic nucleotide sequences. If we see here, it is GAATTC. Here as well, it is GAATTC from both sides. So it can cut here as well as here. Palindromic nucleotide sequences. As against a word palindrome, wherever the same word is read in the both directions, the palindrome in DNA is a sequence of base pairs that read same on two strands when orientation of reading is kept the same. For example, the following sequence reads the same on the two strands. 5' to 3' direction is also true if read in 3' to 5' direction. GAATTC, GAATTC. Okay, restriction enzymes cut the strand of DNA a little away from the center of the palindrome sites, but between the same two bases on the opposite strands. This leaves single-stranded portions at the ends. There are overhanging stretches called as sticky ends on each strand. These are named so because they form hydrogen bonds with their complementary cut counterparts. This stickiness of the end facilitates the action of the enzyme DNA ligase. Restriction endonucleases are used in genetic engineering to form recombinant molecules of DNA which are composed of DNA from different sources. Genomes. When cut by the same restriction enzyme, the resultant DNA fragments have the same kind of sticky ends, and these can be joined together end to end using DNA ligases. Because if we cut using same restriction enzyme, here also same kind of sticky end, meaning they are complementary. Here it is A, here it is T. So AT, double hydrogen bond possible. Adenine pyrimidine bonding. Diagrammatic representation of recombinant DNA technology. foreign DNA, same restriction enzyme for cutting both fragment DNA and vector DNA at specific point. We cut like this here. We cut for like this here, and this enzyme we take it and we include it here because there are sticky ends there. It will get attached ligases, and even ligases help join foreign DNA to the plasmid recombinant DNA molecule. After addition, there is a blue here, if you can see. Blue, this all is red, and there is a blue foreign DNA attached, and this is given to the cells and transformation. Cells divide, cloning host. This DNA will completely get replicated. Recombinant DNA technology. You may have realized that normally, unless one cuts the vector and is source DNA with same restriction enzyme, the recombinant vector molecule cannot be created. Okay, because it needs to have the same complementary sticky ends for them to stick or get attached. Otherwise, it is not possible, and that's why we need the same endonuclease enzyme, meaning same restriction endonuclease enzyme. Separation and isolation of DNA fragments. Separation and isolation of DNA fragments. The cutting, how that is done. Separation, how we are separate and isolate the DNA, required DNA fragments. The cutting of DNA by restriction endonucleases results in fragments of DNA. These fragments can be separated by a technique known as gel electrophoresis, which we separate because we know DNA is negatively charged. We can separate it through density or like mass. You will see the technique, I believe. Yeah, this is the just gel electrophoresis technique. Separation and isolation of DNA fragments, the cutting of DNA by restriction endonucleases. Just we cannot cut the DNA only at one place. That is not possible because the enzyme is a macro molecule and we can't use very minute amounts to cut only one particle of the enzyme to cut at a single spot of DNA. As such, when we use the restriction endonuclease, there will be many cuts at many places, and the whole DNA will be cut wherever there is a palindromic or a recognition sequence. Everywhere there is a recognition sequence, the DNA will be cut. Gel electrophoresis technique used to separate the and isolate the fragments of DNA. Since DNA fragments are negatively charged, they can be separated by forcing them to move towards the anode that is positively charged under an electric field through a medium matrix. Nowadays, the most commonly used matrix is agarose, which is a natural polymer extracted from seaweeds. The DNA fragments separate, resolve according to their size through the sieving effect provided by the agarose gel. Hence, smaller the fragment size, the farther it moves. Look at the figure 9.3 and guess at which end of the gel the sample was loaded. Okay. Separation and isolation of DNA fragments. The cutting of DNA by restriction endonucleases results in fragments of DNA. The fragments can be separated by a technique known as gel electrophoresis. Since what is this gel electrophoresis technique is, DNA fragments are natively charged, so we place them in an electric field and in a medium or a matrix. The most commonly used matrix is an agarose gel, which is natural polymer. It's extracted from seaweeds. DNA fragments separate according to the size through a sieving effect provided by the agarose gel. And the smaller the fragment size, farther the movement. Guess at which end of the sample was loaded. It was at this end. They have a wells here. The sample was loaded. Basically, DNA was loaded at this end, and this is the anode, positive charge, negative charge. Because DNA is negatively charged, the DNA will move this side. Because of that, the smallest DNA is here. A typical agarose gel electrophoresis showing migration of undigested and digested set of DNA fragments. Lane 1 is undigested. Undigested meaning it was not restriction and endonucleases was not used on the first lane of enzymes. Mean it was not cut. That is why there are no fragments there. It's a single strand of DNA. But in 2, 3, 4, the DNA was cut. The separated DNA fragments can be visualized only after staining the DNA with a compound known as ethidium bromide, followed by exposure to UV radiation. You cannot see pure DNA fragments in visible light and without staining. You can see bright orange colored bands of DNA in a ethidium bromide stained gel exposed to UV light. The separated bands of DNA are cut out from the agarose gel and extracted from the gel piece. This step is known as elution. The DNA fragments purified in this way are used in constructing recombinant DNA by joining them with cloning vectors. Okay, the separated DNA fragments can only be visualized after staining the DNA with a compound known as ethidium bromide. Important to remember, the DNA fragments cannot be visualized normally, so they need to be stained by a compound that is known as ethidium bromide, followed by UV light exposure. You cannot see pure DNA fragments in the visible light. First, for us to see the DNA fragments as orange bands, we need to first stain it with ethidium bromide, and we need to use UV exposure. UV exposure, you can see bright orange colored bands of DNA in ethidium bromide stained, gel exposed to UV light. The separated bands are cut out from the agarose gel and extracted from the gel piece. These separated strands will be extracted from the gel piece. Since they are separated now, we will cut them. We will separate them, extract them from the gel piece. And how do we know which is our desired character? After extraction, based on their calculation, the scientist calculation of molecular weight, or I believe molecular weight or something like that, they will know which is the desired character, desired DNA. Extracted from the gel piece. Extraction from the gel piece is known as elution. The DNA fragments purified in this way are used in construction of recombinant DNA by joining them with cloning vectors or plasmids. Cloning vectors, you know that plasmids and bacteriophages have the ability to replicate within bacterial cells and independent of the control of the chromosomal DNA. Bacteriophages, because of their high number per cell, have very high copy numbers of their genome within the bacterial cells. Some plasmids may have only 1 or 2 copies per cell, whereas others may have 15 to 100 copies per cell. Their numbers can even go higher. If you were able to link an alien piece of DNA with bacteriophage or plasmid, we can multiply its number equal to the copy number of plasmid or bacteriophage. Vectors used at present are engineered in such a way that they help easy linking of foreign DNA and selection of recombinants from non-recombinants. Okay, let's read that again. We know that plasmids and bacteriophage have the ability to replicate within the bacterial cells independent of the control of the chromosomal DNA. Okay, information on known. Second, bacteriophages, because of their high number per cell, have very high copy numbers of their genome within the bacterial cell. We know that bacteriophages are viruses that attack the bacteria, and they have very high number copies of their genome. They use a cells machinery, bacteria cells machinery, to make copies of themselves. Some plasmids, bacterial cells, bacteriophages is over. Now, plasmids may have only one or two copies per cell, whereas others may have 15 to 100 copies per cell. Some bacterial cells have two or three plasmids, or like plasmids be only one or two copies per cell, but in some other bacteria, there might be 50 to 100 copies per cell. And knowing that bacteria are microorganisms and there are already billions of bacteria in a small medium itself, the numbers plasmids is very high. The number can go even higher than 100 copies per cell. If you are able to link an alien piece of DNA with a bacteriophage or a plasmid DNA, we can multiply its number equal to the copy number of the plasmid or the bacteriophage. Vectors now used at the present are engineered in such a way that they help easy linking of foreign DNA and selection of recombinants from non-recombinants. First thing what they are saying that vectors used at the present are engineered in such a way that they help easy linking of foreign DNA. They can easily link foreign DNA and selection of recombinant from non-recombinant types. Easy linking of foreign DNA. Older plasmids were natural and didn't have convenient places to insert DNA. Now vectors are engineered, man-made, modified, so they have multiple cloning sites, MCS or polylinkers, short DNA sequence containing recognition sites for several restriction enzymes that gives scientists flexibility to cut both plasmid and foreign DNA with same enzyme and join them easily. Ligase-friendly ends, sticky ends are often preferred for easier joining. This makes it easier to insert a foreign gene into the vector. Selection of not every plasmid will successfully take up the foreign DNA. After transformation into bacteria, there will be recombinants, plasmids with foreign DNA inserted, non-recombinants, plasmids without foreign DNA, empty vectors. To distinguish between them, vectors are engineered with selectable marker genes, antibiotic-resistant genes. Bacteria with plasmid survive on antibiotic plates, while those without it die. Reporter genes like LAG-Z gene, insertion of foreign DNA disrupts the gene, so recombinant colonies can be visually distinguished. Blue-white screening. This allows scientists to easily identify which actually have the desired recombinant. Okay, let's see here. The following are the features that are required to facilitate cloning into a vector. Origin of replication, ORI sites. This is the site that the foreign DNA should actually link to. In chromosomes, there are origin of replication sequences. This sequence, where replication starts, and any piece of DNA, when linked to this sequence, can be made to replicate within the host cells. This sequence is also responsible for controlling the copy number of the linked DNA. So if one wants to require many copies of the target DNA, it should be cloned in a vector whose origins support high copy number. Selectable marker. In addition to ORI, the vector requires a selectable marker which helps in identifying and eliminating non-transformants and selectively permitting the growth of the transformants. Transformation is a procedure through which a piece of DNA is introduced in a host bacterium. We'll study the process in subsequent section. Normally, the genes encoding resistance to antibiotics such as ampicillin, chloramphenicol, tetracycline, or canamycin, etc., are considered useful selectable markers for E. coli. The normal E. coli cells do not carry resistance against any of these antibiotics. E. coli cloning vector PBR322 showing restriction sites of, meaning it has the recognition sequences of all these restriction enzymes. In 3, E. coli 1, BAMH1, SALH, LSALI, PVU2, PST1, CLA1, ORI, and antibiotic resistant genes. It also has ampicillin and tetracycline, ROP codes for the proteins involved in the replication of the plasmid. Okay, let's see the origin of replication again. This is a sequence where replication starts, and okay, that much we know. Sequence is responsible for controlling the copy number of linked DNA. So if you want high number of copies, we need to take a vector whose origins support high copy number. Okay, new information. Second is a selectable marker. In addition to the origin of replication, the vector requires a selectable marker, and the selectable marker which helps in identifying the and eliminating non-transformants and selectively permitting the growth of transformants. We need, after as we saw before, if we did insert recombinant DNA as bacteria are numerous, only some might actually get transformed. Like only some might gain the transformation, like some might gain the altered genes phenotype, others might not have. So for identifying the non-transformant which did not have the acquired phenotype, they needs to be eliminated. We don't need use for them. We have no use for them. We only have use for transformants, that is which have the new phenotype or which have the new altered gene. Selectively permitting the growth of the transformants. Transformation is a procedure through which a piece of DNA is introduced in. What is transformation? Procedure of introducing a recombinant DNA into host bacterium. Normally, genes encoding resistance to antibiotics. These genes encoding resistance to antibiotics are used as selectable markers for E. coli. E. coli doesn't have antibiotic resistance for any of these mentioned antibiotics here, that is ampicillin, chloramphenicol, tetracycline, and canamycin. So it doesn't have any resistance to this. So if normal non-transformed E. coli were to sit on these plates of antibiotics, it will die. But if transformed one had this recombinant gene that codes for resistance against them, then it will live. So that way we can isolate or select transformed E. coli and leave the untransformed one. Cloning sites. In order to link the alien DNA, the vector needs to have very few, preferably single recognition sites for the commonly used restriction enzymes. Presence of more than one recognition site within the vector will generate several fragments, which will complicate the gene cloning. Okay. In order to link the alien DNA, the vector needs to have very few, preferably a single recognition site. Recognition site is the sequence where the endonuclease will attach itself and cut the DNA. So it should have only one recognition site per restriction nucleus, so that it doesn't, we only need, if we want to cut this is a vector, this is circular vector. If we want to add DNA, we need to cut it here and separate it like this and add a new DNA here and rejoin them. But what if there was another restriction then site here and here and here? It will be cut in so many places. It will not be circular anymore. So to avoid that, the vector should preferably have only one recognition site, only one place where the restriction endonuclease can cut. Presence of more than one recognition site will generate several fragments, which will complicate the gene cloning. The ligation of alien DNA is carried out at a restriction site present in one of the two antibiotic restriction genes. For example, you can ligate a foreign DNA at BAMHI site of tetrasilin resistance gene in the vector PBR322. The recombinant plasmids will lose tetrasilin resistance due to insertion of foreign DNA, but can still be selected out from non-recombinant ones by plating the transformants on ampicillin containing medium. Okay. Okay. The this plasmid vector has this plasmid has two antibiotic resistant genes. One is for ampicillin, one is for tetrasilin. These antibiotic resistant genes are the place which also have the recognition sequence. So if endonuclease attacks, if a specific endonuclease, for tetrasilin example, BAMHI, if this will attack, it will remove the tetrasilin gene and it will attach the desired gene character. If it was only one antibiotic gene, then we won't be able to distinguish between non-transformant and transformant. But because it has two antibiotic resistance gene, we are able to find out which have the vector, because this vector we are transplanting into the cell, not something the cell has. So by doing this, we are able to identify which has the cell, like which of the bacteria have the cell, like have the vector transplanted, by taking them to an ampicillin medium, like ampicillin medium, where if it has this vector, because it has the ampicillin gene and the desired DNA was in the tetrasilin site, because of that this will still be there, so it will be resistant and it will not die. But non-transformants which did not have this vector will die. And we can also identify by taking them to tetrasilin, whichever dies has the foreign DNA, which doesn't die is still having the vector. This is for testing if it was successfully transplanted or transferred or not, transformed or not. The transformants growing on ampicillin containing medium are then transferred on a medium containing tetrasilin. The recombinants will grow in ampicillin containing medium, but not on that containing tetrasilin because it was removed and a new recombinant DNA was added. If it was only the vector that was getting like transferred or yes transferred, then it will have both tetrasilin resistance and ampicillin resistance. But this tetrasilin was removed and a new altered DNA was added here. So because of that, this vector doesn't contain tetrasilin resistance. So when the bacteria will grow in ampicillin containing medium because they have antibiotic resistance, but they won't grow on tetrasilin resistance. I mean tetrasilin antibiotic because they don't have that resistance. But non-recombinants will grow on the medium containing both the antibiotics. They are transformants, but they are not recombinants. There is no recombinant DNA here. If they grow on tetrasilin, meaning the resistance to tetrasilin medium, they just got transformed. They were added this vector plasmid. That's all. They did not have the DNA added, the altered DNA. So they are non-recombinants if they grow on both ampicillin and tetrasilin mediums. But if they only grow on ampicillin and die at tetrasilin mediums, then they are transformants and recombinants. Those who do not grow on ampicillin or tetrasilin mediums are called as non-transformants, non-recombinants. In this case, one antibiotic resistant gene helps in selecting transformants, whereas the other antibiotic resistance gene gets inactivated due to insertion of alien DNA and helps in selection of recombinants. Selection of recombinant due to inactivation of antibiotics is a cumbersome procedure because it requires simultaneous spreading of two plates having different antibiotics. Therefore, alternative selectable markers have been developed which differentiate recombinants from non-recombinants on the basis of their ability to produce color in the presence of a chromogenic substrate. This selection of recombinants through inactivation of antibiotics is a hard process because it requires simultaneous plating. A 2 minutes break. Okay, then selection of recombinants due to inactivation of antibiotics is a very hard process because it requires simultaneous plating of two plates having different antibiotics. Therefore, alternative selective markers have been developed which differentiate recombinants from color, from their ability to in produce color in presence of chromogenic substrate. In this, a recombinant of DNA is inserted with coding sequence of an enzyme beta-galactosidase. This results into activation of the gene for synthesis of this enzyme, which is referred to as insertional inactivation. Okay, they're saying that in this, a recombinant DNA is inserted with a coding sequence of an enzyme beta-galactose. This results in inactivation of the gene for synthesis of this enzyme. Basically, or there is a beta-galactose enzyme coding sequence of this enzyme, and DNA, recombinant DNA or altered or required DNA is inserted into the gene sequence coding for the beta-galactose enzyme. So this results as as if we insert the new sequence, this beta-galactose and DNA sequence will be changed, so it cannot code for this anymore. This results in the inactivation of the gene synthesizing for the beta-galactose. This is referred to as insertional inactivation. When we inserted the foreign DNA, the enzyme got inactivated. The production of the enzyme got inactivated. The presence of a chromogenic substrate gives blue-colored colonies if the plasmid in the bacteria does not have an insert. Presence of insert results into insertional inactivation of the beta-galactose gene. The colonies do not produce any color. These are identified as recombinant colonies. Okay. If there is the enzyme, if there is presence of the beta-galactose creating enzyme, then the colonies will give a blue color. But if the plasmid in the bacteria does not have an insert, meaning if it does not have an insert, then blue colored colonies will be produced, meaning beta galactose enzyme is still there. So the transformation was not successful, or I mean recombination was not successful. So non-recombinants which are blue color. And if there is a presence of an insert, there is a foreign DNA that was inserted, then in presence of the chromogenic substrate, the colonies should not be blue in color. They should not produce any color because there is no presence of the beta galactose gene. Do not produce any color. These are identified as recombinant colonies. Vectors for cloning genes in plants and animals. You may be surprised to know that we have learned the lesson of transferring genes into plants and animals from bacteria and viruses, which have known this for ages. How to deliver genes to transform eukaryotic cells and force them to do what the bacteria want. I mean, bacteria or viruses want. Basically, viruses, we know that they are inert organisms. They're acellular organisms. They do not have a cell. They're just floating RNA or DNA with a protein code. They attach to cells, transfer their RNA or DNA, and make use of the cell's mechanism to replicate themselves. This is what they do. So we learned the lesson of transferring genes, that is anything we want, in plants and animals from bacteria and viruses which have known this technique for ages. How to deliver genes to transform eukaryotic cells and force them to do what the bacteria or virus want. For example, Agrobacterium tumefaciens, pathogen of several dicot plants, is able to deliver a piece of DNA known as tDNA to transform normal plant cells into a tumor and direct these tumor cells to produce the chemicals required by the pathogen. Similarly, retroviruses in animals have the ability to transform normal cells into cancerous cells. A better understanding of the art of delivering genes by pathogens in the eukaryotic host has generated knowledge to transform these tools of pathogens into useful vectors for delivering genes of interest to humans. Okay, basically, Agrobacterium tumefaciens. It's a special pathogen of several dicotyledonous plants. It's able to deliver a piece of DNA known as tDNA, transformation DNA, to transform normal plant cells into a tumor. Basically, this bacteria will attach to plants. It will it will transfer a DNA known as tDNA into the plant cells. This tDNA will transform the plant cells into a tumor and direct these plant cells, tumor cells, to produce chemicals required by the pathogen. The pathogen requires several chemicals, and these tumor cells, which have been transformed, will produce those chemicals. Even retroviruses in animals have the ability to transform cells into cancerous cells. Better understanding of this art of delivering genes by pathogens in their eukaryotic hosts has generated knowledge to transform these tools of pathogens into useful vectors for delivering genes of interest in humans. The tumor indicating TI plasmid of Agrobacterium tumefaciens has known to be modified into a cloning vector for which no more pathogenic plants but is still able to use the mechanisms to deliver genes of our interest into variety of plants. Okay, the tumor-inducing plasmid of the Agrobacterium tumefaciens. It used to be a tumorous, dangerous pathogenic DNA, but now the tumor-inducing plasmid of Agrobacterium, this DNA, the tDNA, is actually a plasmid. That is the reason it can persist and change them into tumorous cells. This Ti plasmid, Agrobacterium tumefaciens, it has been modified into a cloning vector which is not pathogenic to plants anymore, and it still uses the mechanisms to deliver genes of our interest into variety of plants. Because it's already a cloning vector that's affecting plants, we can also use it to add a recombinant DNA to it and make several genes deliver genes of several of our interest into the variety of plants. Similarly, retroviruses have also been disarmed and are now used to deliver desirable genes into animal cells. So once a gene or a DNA fragment has been ligated into a suitable vector, it is transferred into a bacterial plant or cell animal host where it multiplies. Okay, once gene or a DNA fragment has been ligated into a vector, we cut the DNA and attach it to a vector. It is transferred into a bacterial plant or animal host where it multiplies. Component host for transformation with recombinant DNA. Since DNA is hydrophilic molecule, it cannot pass through cell membranes. Why? In order to force bacteria to take up the plasmid, the bacterial cells must first be competent to take up the DNA. This is done by treating them with a specific concentration of divalent cation, such as calcium, which increases the efficiency with which DNA enters the bacterium through pores in its cell wall. Recombinant DNA can then be forced into such cells by incubating the cells with recombinant DNA on ice, followed by placing them briefly at 42 degrees heat shock and then putting back on ice. This enables the bacteria to take up the recombinant DNA. This is not the only way to introduce alien DNA into host cells. In a method known as microinjection, recombinant DNA is directly injected into the nucleus of an animal cell. Another method suitable for plants, cells are bombarded with high-velocity microparticles of gold or tungsten coated with DNA in a method known as biolistics or gene gun. Ballistics, biolistics, or a gene gun. And the last method used, disarmed pathogen vectors, which when allowed to infect the cell, transfer the recombinant DNA into the host. Now that we have learned about the tools for constructing recombinant DNA, okay, we have a problem. We have already discussed about how RNA can be recombinant vectors used, etc. But how can this DNA, because it's a hydrophilic molecule and cells having cell wall and cell membrane and such, cannot allow the DNA inside. They are not very welcoming. So how can we take this alien DNA vector inside the cell? Only inside the cell can it replicate inside the cell cytoplasm. So we need to take it inside. But how can we do that? There are various methods to do it. One method is treating them with a specific concentration of a divalent cation, a plus 2 cation, such as calcium, which increases the efficiency with which DNA enters the bacterium through the pores in the cell wall. Okay, that is first method. First method is treating the cell with calcium, a divalent cation, which will increase the efficiency of DNA taken up through the pores in the cell wall. Second method, recombinant DNA can be forced into such cell by incubating cells with recombinant DNA on ice. Second method is we incubate the DNA, recombinant DNA with the cells on ice, then heat it to 40 degrees, then again cool it back. This enables bacteria to take up the recombinant DNA. This is called as heat shock. Third method is known as microinjection. Okay, through the pores, this seems to be a one method. Recombinant DNA can then be forced by, first we use a divalent cation to increase the absorption of DNA through the pores. Then recombinant DNA can be forced by incubating it with the cells in ice and heating it, then again incubating it on ice. And second method is to introduce DNA, recombinant DNA into host cells is microinjection, where we directly use microinjection to inject it into the nucleus of an animal cell. Third method, suitable for plants, is cells are bombarded, like we shoot them or collide them with high velocity microparticles of gold or tungsten. They'll collide with the high velocity particles of gold or tungsten coated with DNA in method known as biolestic or gene gun. This gold or tungsten will be coated with recombinant DNA, and when they collide with the cell, they will enter the cell, and now the cells will have the recombinant DNA. And the last method uses disarmed pathogen vectors. They will use pathogen vectors like bacteriophages, etc., which, when allowed to infect the cell, transfer the recombinant DNA into the host. They will be disarmed, meaning they will not be pathogenic. They will be safe now. Now that we have learned about tools for constructing recombinant DNA, let us discuss the process of facilitating recombinant DNA technology. Okay, let's go to the last topic. Process of recombination Processes of recombinant DNA technology. Recombinant DNA technology involves several steps in specific sequence, such as isolation of DNA, fragmentation of DNA by restriction endonucleases, isolation of desired DNA fragment, ligation of the DNA fragment into a vector, transferring the recombinant DNA into the host, culturing the host cells in medium at large scale, and extraction of desired product. Let us examine each of these steps in some detail. I'll read that again. Let's see the picture here. DNA that separates out can be removed by spooling. This seems to be DNA. Finally, that be. Let's read this again. Recombinant DNA technology involves several steps in specific sequence, such as isolation of DNA. First step is isolation of DNA, fragmentation of DNA by endonucleases, isolation of desired DNA fragment, which we saw through gel electrophoresis, ligation of DNA fragment into a vector. We attach it to a vector, that is a plasmid, transferring recombinant DNA into the host. Now transfer vector into a host. This process also we saw how it's done. We saw gene guns, biolistics, incubation, calcium divalent ions, microinjection method, and so on, or even using pathogens which are disarmed. Okay, now let's see isolation of genetic material DNA. Recall that the nucleic acid is a genetic material of all organisms without exception. Nucleic acid is the genetic material of all organisms without exception. In majority of organisms, this is deoxyribonucleic acid or DNA. Okay, nucleic acid is the genetic material of all organisms without exception. Well-known knowledge, well-known information. In majority of organisms, this is deoxyribonucleic acid or DNA, also known. In order to cut the DNA with restriction enzymes, it is needs to be in pure form, free from other macromolecules. Okay, if you want to cut the DNA using restriction endonucleases, there should only be DNA, no other proteins or anything else, other macromolecules. It should only be DNA. Since DNA is enclosed within the membranes, we have to break the cell open to release DNA along with other macromolecules such as RNA, proteins, polysaccharides, and also lipids. The cell contains the nucleus. Nucleus contains the DNA. There are many organelles, many membranes which cover the DNA. And to release the DNA, we have to break the cell open, release DNA along with other macromolecules such as RNA, proteins, polysaccharides, and also lipids. This can be achieved by treating the bacterial cell plant or animal tissue with enzymes such as lysozyme bacteria, cellulase plants, chitinase fungus. Okay, if you want to break the cell open, we need to use lysozyme for bacteria, cellulase for plants to remove cellulose membrane, I mean cellulose which is making up the cell wall, and chitinase for fungus because fungus have chitin. You know that genes are located on the long molecules of DNA as sequences intertwined with proteins such as histones. The RNA can be removed by treatment with ribonuclease, whereas proteins can be removed with treatment with proteases. We remove RNA with the help of a ribonuclease enzyme, RNase enzyme, and protease enzyme will remove proteins like histone proteins, which wound up the DNA. Other molecules can be removed by appropriate treatments with purified DNA ultimately precipitates out of the addition of chilled ethanol. This can be seen as collection of fine threads in the suspension figure 9.5. Okay, this here is chilled ethanol here. After purification, if we add chilled ethanol, it will appear as threads like this and it will come out easily. It will precipitate out. Cutting off DNA at specific locations. Restriction enzymes digestions are performed by incubating purified DNA molecules. Okay, before that, let's revise here once again. First, we break open the cell using lysozyme for bacteria, cellulase for plant cells, and chitinase for fungus. Now we remove RNA of the cell by RNases or ribonucleases, proteins by proteases. Then we precipitate DNA out through a chilled ethanol solution. It will come like a free suspension shown here. Now cutting DNA at specific locations, restriction enzyme digestions or cut are performed by incubating purified DNA molecules with restriction enzyme. Okay, we incubate this DNA with restriction enzyme so that it will act at the optimal condition for the specific enzyme. Agarose gel electrophoresis is employed to check the progression of restriction enzyme digestion. Agarose gel electrophoresis is employed to check the progression of a restriction enzyme digestion. To know if the digestion is going on or not, we use a gel electrophoresis to separate the fragments. If we get fragments, we know that it is the gel electrophoresis, I mean the digestion is going on. If we do not get fragments, it does not get separated, then we know that the restriction endonucleases are not working. DNA is a negatively charged molecule, hence it moves towards the positive electrode anode. The process is repeated with vector DNA as well. We know that we use same restriction endonuclease on vector as well, so that we have sticky ends, and it is repeated with vector as well. The joint DNA involves several proteases. After having cut the source DNA as well as the vector DNA with specific restriction enzyme, the cut out gene of interest from the source of DNA and cut vector with space are mixed and ligase is added. After cutting the source DNA as well as the vector DNA, both will cut using the restriction endonucleases. The cutout gene of interest from the source of DNA, we will separate the DNA. After the cut DNA, the DNA will be cut after the separation from the gel electrophoresis. We will separate it. The gene of interest, that DNA, the DNA having the gene of interest, and the vector, the cut vector, will be added together along with a ligase to get them mixed, to get them attached, and ligase is added. This results in the preparation of recombinant DNA. Now, amplification of gene of interest using PCR. PCR is known as polymerase chain reaction. In this reaction, multiple copies of the gene or DNA of interest is synthesized in vitro using two sets of primers. Let's read first. Two sets of primers. Small chemically synthesized oligonucleotides that are complementary to the regions of the DNA, and the enzyme DNA polymerase. The enzyme extends the primers using the nucleotides provided in the reaction and the genomic DNA as template. If the process of replication of DNA is replicated many times, the segment of DNA can be amplified to approximately billion times, that is 1 billion copies are made. Such repeated amplification is achieved by the use of thermostable DNA polymerase isolated from a bacterium Thermus aquaticus, which remains active during high temperature. Induced denaturation of double-stranded DNA. The amplified fragment is desired can now be used to ligase with vector for further cloning. Okay, basically why we need this PCR is because we need more gene of interest. We separated the DNA fragments. Now for our separated gene of interest, we need more of it, more copies of this gene of interest, so that we can add more vectors to it and we get more plasmids which are recombinant and more bacteria which can get transformed and become recombinant, so that we need a way to artificially make many copies of desired gene of interest. To do that, we have PCR, polymerase chain reaction. In this reaction, multiple copies of gene or DNA of interest are synthesized in vitro. Billions of copies are synthesized using two primers. Let's see the diagram here. First is there is a double-stranded DNA here, or DNA of interest, double-stranded DNA here. We heat it, then primers get attached at the 3' to 5' end. Annealing, denaturation, annealing, deoxynucleotides, the cycle amplified billion times. Okay, I understand. There is a DNA here. After heating it, the bonds will be broken. The bonds will be broken here. These bonds will be broken since DNA is a double helix. After heating, these bonds will be broken. That's why we have a separation here. Primers are actually artificially created oligonucleotides. Nucleotides, basically base pairs, which just act like the complementary strand. After this separation, this is one strand, this is a single strand. Now, primers, which are basically nucleotides, will get attached here like this. A single part of the DNA, they'll get attached here on both segments. Now, we add DNA polymerase and deoxynucleotides. DNA polymerase, from molecular basis of inheritance, we know it will start extending these nucleotides like this, like this, and a new chain is formed. A new complementary strand is formed. This is like DNA replication, artificial DNA replication, polymerase chain reaction. With this, we get two new strands of DNA like this. And the same cycle we repeat, we do it for 30 cycles, amplification a billion times, 10 power 9. Sets of primers, small chemically synthesized oligonucleotides that are complementary to the regions of DNA, and the enzyme DNA polymerase. The enzyme extends the primers using nucleotides, providing in reaction and genomic DNA as template. The process of replication of DNA is repeated many times. The segment of DNA can amplify to approximately billion times. 1 billion copies are made. Such repeated amplification is achieved by the use of thermostable DNA. We know that normal enzymes, normal enzymes, are unable to work at very high temperatures, unable to work properly at very high temperatures. Because of that, we use a very thermostable DNA polymerase, an enzyme which is very thermostable, and it is isolated from a specific bacterium, Thermus aquaticus, which remain active. This enzyme will remain active even during high temperature. Induced denaturation of double-stranded DNA through heating. The amplified fragment, if desired, can now be used to ligate with a vector for further cloning. Now, after this DNA, we will add this to a vector and you will get cloned. Insertion of recombinant DNA into the host cell. There are several methods for introducing ligated DNA into recipient cells after making them competent to receive, take up DNA present in its surroundings. So if a recombinant DNA bearing gene for resistance to antibiotic, example ampicillin, is transferred to E. coli, the host cells become transformed into ampicillin resistant cells. If we spread the transformed cells on agar plates containing ampicillin, only transformants will grow, simply similar to selectable markers which we saw. Untransformed recipient cells will die, where the selectable markers there we saw that this was a too cumbersome process for a selectable marker because plating them with antibiotic is very hard to do. Because of that, we use beta galactose enzyme. So if there is a beta galactose enzyme, then it will be blue color. No beta galactose enzyme after insertion of a or alteration of the DNA by addition of a foreign DNA, which is recombinant type. No color if it is recombinant type, color if it is blue color if it is non-recombinant type. Since due to ampicillin resistant gene, one side is able to get transformed cell in the presence of ampicillin. The ampicillin resistant gene in this case is called as a selectable marker. Obtaining a foreign gene product. When you insert a piece of alien DNA into a cloning vector and transfer it into a bacterial, plant, or animal cell, the alien DNA gets multiplied. In almost all recombinant technologies, the ultimate aim is to produce a desirable protein. Hence, there is a need for recombinant DNA to be expressed. The foreign gene gets expressed under appropriate conditions. The expressions of foreign genes in host cells involve understanding many technical details. After having cloned the gene of interest and having optimized the conditions to induce the expression of target protein, one has to consider producing it on the large scale. Can you think of any reason why there is a need for large-scale production? If any protein encoding gene is expressed in a heterologous host, it is called a recombinant protein. The cells harboring cloned genes of interest may be grown on a small scale in the laboratory. The cultures may be used for extracting the desired protein and then purifying it by using different separation techniques. The cells can be amplified. Okay, not much summary here. Let's read that again. Obtaining the foreign gene product. When you insert a piece of alien DNA into a cloning vector and transfer it into a bacteria, plant, or animal cell, the alien DNA this gets multiplied. Okay, known information. In almost all recombinant technologies, the ultimate aim is to produce a desirable protein. We need to produce a protein. That is a desirable goal here. Hence, there is a need for recombinant DNA to be expressed. If it is expressed only, we can get the desirable protein. If it is not expressed, it means it's non-recombinant. The foreign gene gets expressed under appropriate conditions. The expression of foreign gene in host cells involve understanding many technical details. After having cloned the gene of interest and having optimized the conditions to induce the expression of the target protein, one has to consider producing. Okay, after having cloned the gene of interest and having optimized the conditions to induce the expression of the target protein, one has to consider producing it on a large scale. We need to produce it on a large scale. Can you think of any reason large scale production like insulin? If we take, it's for diabetic patients, it's utterly important, very important. It's impossible to survive without insulin. It's very hard. If any protein encoding gene is expressed in a heterologous host, it is called a recombinant protein. Excuse. So they're saying that if any protein encoding gene, it's a protein encoding gene, a recombinant gene, is expressed in a heterologous host, whatever that means. Alien DNA is multiplied in a host cell. When you insert foreign DNA into a vector and put it into a host cell, they replicate along the host machinery. So you get many copies. In biotechnology, the aim is often not just copy DNA, but make a specific protein coded by the DNA. Example, the insulin gene coded into E. coli bacteria make human insulin. Hepatitis B surface antigen in yeast make the vaccine protein. Expression of foreign gene. Expression means that DNA is actually used by the host cell to produce mRNA protein. For this to happen, scientists must ensure the gene is placed under a promoter that works in the host conditions, temperature, nutrition, etc. allow high level expression. While large scale production in lab, can you grow small flasks of cells and purify tiny amounts of protein, but for medical or industrial use, insulin for millions of diabetics need large quantities that requires large scale scaling up of bioreactors with thousands of liters of culture. Recombinant protein, if a protein coding gene is expressed in a heterologous host, a different organism than its natural source. Heterologous host means a different organism than its natural source. A protein made is called as a recombinant protein. Example, human insulin made in E. coli is a recombinant protein, but because bacteria naturally don't make insulin, protein purification after cells produce protein, it must be extracted and purified. Different separation techniques are used: centrifugation, chromatography, electrophoresis, etc. This ensures the final protein is pure, safe, and usable for therapy or industry. In short, recombinant DNA technology isn't just about cloning DNA, it is about expressing the DNA to make useful recombinant proteins like insulin, vaccines, enzymes, and then producing them at a large scale so that they can be used in medicine and industry. Okay, The cells can also be multiplied in continuous culture system wherein the use of medium is drained out from one side with fresh medium is added from the other to maintain the cells in their physiologically most active log exponential phase. The cells can be multiplied in a continuous culture system wherein the used medium is drained out from one side with fresh medium is added from other to maintain the cells in their physiologically most active state. This type of culture method produces larger biomass leading to higher yields of desired protein. Small volume cultures cannot yield appreciable quantities of products. To produce in large quantities, the development of bioreactors, where large volumes, hundreds to thousands of liters of culture can be processed, was required. Thus, bioreactors can be thought of as vessels in which raw materials are biologically converted into specific products, individual enzymes, etc. Using microbial plant, animal, or human cells, a bioreactor provides optimal conditions for achieving the desired product by providing optimal growth conditions, temperature, pH, substrate, salts, vitamins, oxygen. The most commonly used bioreactors are of stirring type, which are shown: simple stir tank bioreactor, B sparged stir tank bioreactor through which sterile bubbles are sparged. It's a foam breaker here, motor, acid base for pH, buffer I believe, control, steam for sterilization, foam breaker, flat blade impeller, culture broth which rotates, sterile increased surface area for oxygen transfer, gas and entrainment, bubbles dramatically increase the oxygen transfer area. A stirred tank reactor is usually cylindrical or with curved base to facilitate the mixing of the reactor contents. Yes, circular because it needs to facilitate mixing. The stirrer facilitates even mixing and oxygen availability throughout the bioreactor. What is the function of the stirrer? It is to mixing and facilitation of oxygen. That is why bubbles here. Alternatively, air can be bubbled through the reactor. If you look at the figure closely, you will see that the bioreactor has an agitator system, an oxygen delivery system, and a foam control system, a temperature control system, pH control system, and sampling ports so that the small volumes of culture can be withdrawn periodically. That is replacing it with new medium. Downstream processing. After completion of the biosynthetic stage, the product has to be subjected through series of processes before it's ready for marketing as a finished product. The processes include separation, purification, which are collectively referred to as downstream processing. The product has to be formulated with suitable preservatives. Such formulation has to undergo thorough clinical trials, as in case of drugs. Strict quality control testing for each product is also required. The downstream processing and quality control testing vary from product to product. That's with the lesson. Let's see the summary. Biotechnology deals with large-scale production and marketing of products and processes using a living organism, cells or enzymes. Modern technology, biotechnology using genetically modified organisms, was made possible only when man learned to alter the chemistry of DNA and construct recombinant DNA. This key process is called as recombinant DNA technology or genetic engineering. This process involves the use of restriction endonucleases, DNA ligases, appropriate plasmid or viral reactors to isolate and vary the foreign DNA into host organism, expression of foreign gene, purification of the gene product, its functional protein, and finally making suitable formulations for marketing. Large-scale production involves use of bioreactors. The key process is called as recombinant DNA technology or genetic engineering. This is the main process that led man to come to the current recombinant DNA technology. Only when man learned to alter the chemistry of DNA and construct recombinant DNA, this key process is called as recombinant DNA technology or genetic engineering. This process involves the use of restriction endonucleases, DNA ligase, appropriate plasmid, and viral vectors to isolate and ferry the foreign DNA into host organisms. Okay, with that, I have completed the first revision of biotechnology and applications. Completed. See you later.